All right, we are on page 16. Now let's just check and make sure. So I'm going to highlight protein because it's going to be a vocabulary word. Why am I highlighting vocabulary words? Because you need to remember them, and you will be making what? You'll be making a study sheet. I'm going to show you the size of paper that you will get for your study sheet. Nice and big. Okay, so you get to use the front of this. You get to use the back of it. If you don't want a big sheet, it is the same size as two of these put together, so you could also use like two smaller sheets. Up to you whether you want the big one or the small one. All right? So by next Friday, your sheet needs to be made. So if you want to start on it this weekend, if you just kind of want to like rough draft things, that's great. If at any point you want the actual piece of paper, let me know. I can grab you one. Okay? But I'm going to do my best to star and highlight any words that should be part of your thing. So you'll notice amino acids is starred and highlighted. Proteins, starred and highlighted. And so what do amino acids make up? Amino acids make up proteins. So they are the building blocks of proteins. Proteins and other living and other chemicals make up living cells. And if you're wondering what page we're on page 16. Okay. Cells make up tissues. And if you're like, what the heck is a tissue? A tissue is, you know, your heart's made of heart tissue, and that's going to be different than your liver, which is made of liver tissue. The cells are slightly different. They have different characteristics. Your bones are made of bone tissue. Yeah, so different genes are expressed in each cell, making all the tissues slightly different. So proteins and other chemicals make up cells. Cells make up tissues. Tissues make up organs. And organs, this should say, and organs, all put together make living creatures. I need to fix that. That star is for me. You can just ignore that star. That means I just need to make a fix there. Okay. Any guesses? How many different kinds of proteins make up our body? Millions of different kinds of proteins make up our body. But in order to function properly, those proteins must be the perfect shape. So you can imagine if you have a nail and you want it to go into a piece of wood, what shape tool would you need? Hammer shape. Yes? That'll be good. But what if you have, instead of a nail, you have a screw? Screwdriver, because if you try and hammer in a screw, it's got all those little things sticking out the side. It doesn't work very well. So in order to do a proper job, you need the proper tool, and tools have the correct shape for their job. Proteins are basically the tools of your body. They do all the jobs that your body needs done, okay? There's a few other things that do things as well, but if you think my body needs to build a cell, well, what's going to help with that building process? Proteins are going to do those jobs. Your stomach says, I need to digest some food. Well, there's enzymes in your stomach that digest the food. And what are those enzymes made of? Protein. Your body says, I need some melanin in my skin so that I don't get burnt by the sun. What do you think that melanin is made of? It's like a pigment. It's like a color. It's made of protein. Okay? So the pigments in your eye, the pigments in your hair, the pigments in your skin, those are all made of proteins. The digestive enzymes are made of proteins. Your muscles that can move are made of proteins. You need a little hormone to signal another part of your body. There are some hormones which are not proteins, but a lot of the hormones are proteins. So basically, if your body needs a job done, who's it going to ask? Who are you going to call? Protein busters. Sorry. All right. One of the functions of the DNA is to tell the amino acids how to line up. So it puts them in the correct order and form themselves into the perfect protein shape. I don't know if you remember these letters from yesterday. A, T, G, and C. Everybody needs to know the letters A, T, G, and C. If you are interested in doing enriched, you can know that A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. So that is optional. Okay, you don't have to memorize those names. You can just say A, T, C, and G. Do you remember who pairs up with whom? A goes with T, at, and C goes with G. The steps, which are also called the rungs of the ladder, are made up of how many kinds of chemicals? How many kinds of chemicals make up the rungs of the ladder? 4. And what are they? What are the letters? A, T, G, C. Excellent. So what we can do is we can look at half of the molecule. So we're going to just fade out 1/2 of the molecule, and we can read it just like we were reading a sentence or a book. And we can say the code goes A, T, G, C, T, A. A, T, G, C, T, A. That's what the code is. And that code is going to tell us which amino acids to put in the string. Okay? So what will happen is you can read the chemical sequence or genetic code from top to bottom. Sort of like a book. Yes.
Just some clarification.
Yes.
Yeah, 2 codes beside each other.
Yep.
2 A's in a row, right?
Yep.
Okay, thank you.
Yeah, so you can have anything beside each other that you want to. I think on the next page it actually says, I don't know if it says, it's going to say it somewhere, but I'm going to repeat it here. Each 3 letters is the code for 1 amino acid. So ATG might say, go get a aspartic acid, okay? And then CTA might say, go get a glutamine, or whatever those fancy names are for amino acids. You don't need to know the names of the amino acids, but you should know that 3 letters is the code for 1 amino acid. Yeah, yeah. Now, a single strand of DNA is extremely long. How many letters long? Millions of letters long. So we're going to zoom out. Okay, so we're starting in at a zoomed in view, and then we're going to zoom out, and we're going to zoom out, and we're going to zoom out some more. How long does that strand look to be? Short or long? Really long. It's as though you have a bunch of yarn and you've just like thrown it in a pile. You took an entire ball of yarn, you stretched it out, and then you threw it in a pile. This long, thin, noodly form, I love the word noodly. I'm sorry, I'm going to say it a lot because noodly is just a great word. It means like a noodle, noodly. The long, thin, noodly form of DNA is called chromatin. And guess what? This is going to be a vocab word. So DNA spends most of its life coiled up like a noodle, living inside the, where does DNA live? DNA lives in the nucleus. Amino acids, however, they live outside the nucleus. Tell your partner if you can remember what we call the area of the cell which is outside the nucleus. Tell your partner if you can remember the area of the cell which is outside the nucleus.
That is close. Well, that's like the outside. We want the outside of the nucleus.
So what's the bulk of the cell made of? Starts with a C. Cyto. And actually, if you want to know the meaning of the prefix cyto, cyto literally means cell. So it means cell plasm. So if you ever hear something about cytokinesis, I don't know if you've heard it. Have you heard of kinetic energy? Kinetic energy is the energy of movement. So kinesis is moving. So you might sometime hear the word cytokinesis. What do you think is moving in cytokinesis? Cells are moving. And they actually do move depending on what you've got. Like bacteria cells can move. Yes? So cytokinesis is cell movement. Cytoplasm is the plasm that's in the cell, and plasm is sort of like a jelly-like substance. Okay, so cyto means cell. So amino acids live outside the nucleus in the cytoplasm. So here's our picture of the cell. There's the nucleus, that's where the DNA lives. Cytoplasm, that's where the amino acids live. And what is the purpose of DNA? Yes. It tells the amino acids how to line up. So we have a problem. Remember, this is like as if I went to IKEA and I brought home the furniture and I took the instructions and I walked them into the house and I put them in the house and I closed the door and then I went back outside to where I've got the parcel in my driveway and I opened the package and I'm like, all right, let's go. Let's assemble this furniture. Yes, my instructions are in the house and I'm out here with all the pieces of furniture. It's not very useful, right? So we're going to have to figure out a way to get those instructions out of the house, out of the nucleus, and into the cytoplasm. And here's the big problem. The DNA, how big is that molecule? Really big. It cannot, there are little tiny holes in the nucleus called nuclear pores, which we'll look at in a little bit. The DNA is too big to get out through those pores. It's just a really big molecule. You cannot get it out of the nucleus. DNA stays in the nucleus. It's actually quite important that it stays there because if it's out and about, it could get damaged. What happens if you get damaged DNA? You get wrong instructions. Those are called mutations, and mutations can lead to all sorts of things. They can do nothing. They can cause cancer. They can actually cause positive things to happen. Sometimes that's how evolution works. Over a long, long period of time, you'll have some good mutations that take hold in a population, right? So you'll have, opposable thumbs, which are very useful for grasping things. All right. So how does the DNA, which is stuck in the nucleus, get its code out to the cytoplasm to help organize the amino acids? Well, what does it do? To help the DNA interact with the cytoplasm, and convert what are we converting into proteins? Amino acids. Into proteins, special chemicals inside the nucleus make, this one's a little hard, partial copies. I don't know why I changed pen color. I just did. Partial copies of the DNA code. You don't want to make a copy of the whole DNA code because then you have a big, big molecule again. So you want to make a copy of just a tiny section because you don't need all the DNA to have the code for one protein. A small little section will have the code for one protein. Yeah? So it's like if you have a big recipe book, all your DNA is the recipe book, and if you go down to one little segment, that's one particular recipe that will make one dish, okay? And the dish is the protein. These partial copies of the DNA code are called RNA, which is short for, yes? Friday. Not this Friday, next Friday, okay? And if you miss it, that means you'll have to come in on the Monday or Tuesday to make it up during, week? I week, okay? Hopefully everyone will make it on that day, but I know people get sick, things happen, right? But yeah. RNA is short for ribo nucleic. That should be in red, shouldn't it? Do you remember why we call it nucleic? Because it's in the nucleus. And then the A stands for acid. And someone pointed this out to me the other day. I can't believe I took a degree in biochemistry and I never noticed. Those of you who did acid naming, yes? Yeah. Those of you who did acid naming, you might notice that this ends in ic. Oh, and I never noticed that. I'm like, it's a nucleic acid because often the names of acids end in ic, like hydrochloric acid, sulfuric acid. And I was like, 52 years old and I learned something new. So cool. Anyway, ribonucleic acid. Do you remember what the D in DNA stands for? And remember, this is an enriched thing. The DNA stands for deoxy ribo. So we got ribonucleic acid and we have deoxy ribonucleic acid. So some chemicals go in the cell and they'll make a little copy. So let's say I wanted to make a copy of this section of DNA here. Some chemicals would go along and be like, let's make a little copy. And that is called RNA. So all those little yellow bits are the RNA. On your page, they'll be gray because everything's gray in your book. I'm so sorry. If I could give you all color books, I would, but it costs about 5 or 6 times as much. So we're not going to do that. All right, here's RNA. What does it look like compared with DNA? Feel free to look back at your molecule of DNA. Tell your partner what's different. What's different about the RNA from the DNA? Yes, Rehan. First, you're going to have to think of something that is different about the RNA and the DNA. Anytime anybody wants to go to the washroom while I'm asking questions, you have to answer my question first. Have a chat amongst you. You can ask someone else if you want to. Aaron. Sorry? It's only 1/2. Yes? DNA is a ladder with rungs that go across. This is like a half a ladder. So we say it is single-stranded. Whereas DNA is, what would we call DNA? Not single-stranded, but double-stranded. Max. Why is there a U? That's another thing that is different. Okay, so you'll notice we have G's. We have C's. And unfortunately, we don't have any A's here, but there could be an A. Here, I'm going to make an A up here. Okay. I'm going to pretend that this one up here is an A, even though I can tell that it's not. Okay, we're going to pretend that this one up here is an A. RNA has U instead of T. We'll talk more about that when we get more into depth in RNA. There are some other differences between RNA and DNA. RNA is short, DNA is long. Yes? Pardon me? Uracil was actually the first of those, we call those nucleotides. We'll come to that word later on. Okay, A, C, G, and T are called nucleotides. Uracil actually came along before thymine. Okay? Because RNA actually came along before DNA. And along the way, the uracil molecule and the thymine molecule, the U and the T molecules, are very similar. There's just a very slight difference in them. There may be 1 or 2 atoms difference. Okay? And it turns out that uracil is more stable. I think it is. Uracil is more stable than thymine, which will allow thymine to undergo some mutations which can lead to evolution. Oh. Anyway, it's quite complicated as to why we actually ended up with thymine instead of uracil in DNA. Technically, you could have uracil in DNA, but we just don't. Okay? So RNA looks a lot like DNA, but they are shorter, of course, and are missing one of the sides. And RNA is what kind of stranded? Single-stranded. So it's not only shorter, but it's also narrower. Half of a ladder is a lot narrower than a full ladder. It's half as wide. Yes? And because it's short and half as wide, here is our cytoplasm. Here is our nucleus. Do you remember what we call this thing that surrounds the nucleus? That is called the nuclear membrane. Is that one of the words that you need to know? Yes. Did I star RNA? I haven't done it yet, so I will put a star beside it and highlight it. And if you had to tell me what a nuclear pore is, tell your partner how you would describe a nuclear pore. What is a nuclear pore? It's just a hole in the nuclear membrane. It's just a hole in the nuclear membrane. So you know like how your skin has pores and those are just little holes in your skin? The nuclear membrane has pores and those are nuclear pores. And because the RNA is short and skinny, it can fit through the nuclear pores. Okay, so RNA is shorter than DNA, it's half as wide in the DNA, and therefore it can fit through the nuclear pores. Isn't that great? So now we're getting the instructions which were written on the DNA. We've made a copy of those instructions, and now that copy is moving out into the cytoplasm, which is like moving out onto your driveway where you have your box of IKEA parts ready to put together. Okay. The RNA molecules, small shape and size, allow them to fit through the tiny what's? Pores in the nuclear, I should have put 2 blanks here. I think I was just going to say nucleus, but the nuclear membrane. And they can fit through there and they go out into the cytoplasm. And from there, once they are in the cytoplasm, they go and find themselves a particle called a ribosome. Remind me, what does a ribosome do? Tell your partner, actually, what does a ribosome do? So I put a star beside ribosome because it is one of your words that you need to know. What do ribosomes make? They make proteins. So a ribosome is a protein-making machine. So what does it do? It reads the RNA code. And by reading that code, it can line up the amino acids in the correct order. It can assemble the amino acids in the correct order. It puts together amino acids in the correct order. So if we're using that analogy where I've got my instructions now out of the house and I'm in my driveway, I would be the ribosome, assembling all the pieces of my IKEA furniture into a proper piece of furniture. And all the pieces of the furniture, those would be amino acids. Ribosomes are what building machines? Protein building machines. They read the RNA code how many letters at a time? 3 letters at a time. And what do they suck out of their surroundings? Amino acids. And they stick them together in a chain according to the genetic code. And the genetic code is the sequence of A's, T's, or U's in this case, C's, and G's. For those of you who want to know the true names, adenines, thymines, cytosines, and guanines. Whoops. Is good? Okay. And remember, you don't need to know all the big long names. You can just go with A, C, T, G. By the way, I have also met, yes, Maya. What are the 4 letters in DNA? Yep. And which 2 go together? Which pairs go together? A's and T's go together, and the C's and the G's go together. Yes, you can go to the bathroom. Okay. As the amino acid chain grows, it bends and it folds. So what's really happening is that this molecule has little bits on it that are positive and negative. And what do positive and negative do? They attract each other. So parts of that amino acid chain are attracting other parts of that amino acid chain. The negative parts are attracting the positive parts and causing it to bend and stick to itself. So if I'm, like, let's just pretend my arms are the amino acid chain. If this elbow was positive and this hand was negative, it would fold like that, yes? It's more sophisticated than that, but that's the general idea. It would fold just like that. And it sticks to itself. To form a perfectly shaped protein. So there's our growing chain of amino acids. It's bending, it's folding, it's sticking to itself. Every how many letters of the RNA code? 3. Every 3 letters of the RNA code tell the ribosome which of the, how many amino acids are there? 20. Different kinds of amino acids should be added next. For example, and I do not expect you to memorize this. Nobody is ever expected to memorize these. Well, maybe in like 4th year university or something like that, but really, there's like a little code book. You can look it up. There's a chart where you can look up and it will say CAA and it will say glutamine. Okay, and it'll say AGU, and beside that it'll say serine. So you can look up which 3 letters code for which amino acid. So CAA tells the ribosome to grab a glutamine. And these amino acids are just floating around, okay? And once again, it's going to grab a glutamine because it's got exactly the right, like, sort of stickiness, sticky areas to grab that. And AGU tells it to grab a serine, and so on. You are not responsible for knowing the names of the amino acids. So there's the ribosome, it's reading the RNA, and it's grabbing these amino acids, and then out of the top part of the ribosome comes the assembled protein. So we're still out in the cytoplasm. We've now created a completed protein. Couple things, a ribosome is actually made of two parts. They do have two different names. You do need to know these names. There is the large subunit and there is the small subunit. Those are their official names. They're not very difficult. Yes, one is bigger and it's called the large subunit. The other part is smaller and it is called the small subunit. Once a protein is built, it can go on to do a number of different things, one of which could be to help form a brand new cell. Ooh, look, there's a cell. And do you see all those cells? They all kind of look like each other. That would form a tissue, okay? So here's a recap. DNA is a molecular blueprint. And if you don't like the word blueprint, you're like, I don't know what a blueprint is. You could say recipe or set of instructions for a living thing. In order to make the protein, we have the DNA, and we make partial copies. What are those partial copies called? RNA. And then the RNA, we read that and we make a protein. So DNA makes RNA, RNA makes protein. And proteins go on to form life. Okay, we got a little bit left here. There's a few more words that we're going to learn. And one of those words is synthesis. So in fact, all of these words, they're all vocabulary words. Except for polypeptide, because that is an optional word. We've already learned protein. So let's just go back up and see what all these words are. Have you guys ever heard the word synthesis before? Kind of.
Photosynthesis.
There's photosynthesis. Do you know what's happening in photosynthesis? You're taking light and you are making
Energy.
sugar.
Sugar.
Okay, we're making usable energy in the form of sugar. Okay, so we are making sugar through photosynthesis, which is making by the process of using light. So synthesis means making or putting together. So has anyone heard the word synthesis in maybe an English class where you have to synthesize ideas? Okay, it's where you take a few ideas and you put them all together and you synthesize a bigger concept. Okay, so that's where you might hear the word synthesize. So synthesis is making. mRNA, there are actually 3 types of RNA that we're going to learn about. One of them is called mRNA. The actual word messenger, you don't have to learn unless you want to, okay? But you can just learn it as mRNA. But the word messenger kind of makes sense because it takes the message of the DNA code from the cytoplasm to from the nucleus to the cytoplasm. So let's go look over here. So in the nucleus, here we have the nucleus. We have the synthesis of the mRNA. What does M stand for? Messenger. So this is no different than what we were looking at before. We make a partial copy of the DNA, and that partial copy is RNA. The only thing we've said now that's different is we have messenger RNA. So is this part familiar? We make a partial copy of the DNA and we call it RNA, or in this case, messenger RNA. What happens to the mRNA now? Right now it's in the nucleus. Where does it go? It goes out into the cytoplasm through, what is this thing here? That is a nuclear pore in the nuclear, what? Membrane. And out goes the messenger RNA. So up here at step 1, we said that's the synthesis of the mRNA. In other words, we are making mRNA. And the process of making mRNA is called transcription. Trans means across, script means writing. Transcription, we're turning the writing of the DNA code, into RNA code. I don't know if you guys remember, like, from, I don't know where you would have learned it, history or something like that. Back in the Middle Ages, there were these monks, yes? And they would sit there and they'd have their little bald head and they would have a copy of some manuscript. They did not have printing presses back then. How would they make a copy of this writing? They would literally sit there and copy it out by hand. And that process of making that copy is called transcribing, which is the same as transcription. So the monk would sit there and transcribe or do the process of transcription by taking one thing and making a physical copy of it. So the process of making an RNA copy of the DNA is called transcription. Just like those monks sitting there and making a copy. So that's another one of our new words, transcription. Making an RNA copy of the DNA. So first, we make the copy. Second, the mRNA moves out into the cytoplasm. Third, we get the synthesis of protein. What does it mean if we are synthesizing protein? We are making protein. And who is making the protein? The ribosome is making the protein. And what is it reading?
The RNA.
It's reading the RNA. What kind of RNA is this? mRNA. It brought the message from the nucleus to the cytoplasm. That's why it's called messenger RNA. It brings the message from the nucleus to the cytoplasm. And the process of making a protein is called translation. Can you think of a time when you might have to translate something? Yes.
Translating languages.
You would have to translate languages, yeah? If you're in French class or Spanish class or whatever language you're taking, you might have to translate from French to English or from English to French. We are translating the RNA code into the protein language. We're reading those 3 letters at a time, and instead of having 3 letters, we're going to have 1 amino acid for each 3 letters. So we're turning that genetic code into a protein code. Now, it is 25 after. I think we should have a 5-minute break, and then we'll come back, and then we'll finish. Okay? 5-minute break. Enjoy yourselves. Back by 2:30.
it's gonna be like vocabulary, knowing what the process is, being able to either like describe the process or try it or something like that. Yeah. That's what it was just asking. Yeah. Yeah, I can take it. Oh, I'm studying, but I know won't be any quizzing because basically instead of quizzing, you're gonna be putting together your like study. Yeah, that's one like a little more. So people who are good at, keeping their homework line, but with weekend coming up and starting making that change, people do well. People who aren't so good at that, and start. After science 10. So if you are signing up for courses.
If there is anyone who is planning on taking Science 10 over the summer and you want more information about grade 11 courses, I know it's not very many people, but there's always one or two. Just let me know, I can give you more information because Science 10 is easy. You sign up for Science 10 or you sign up for Science 10 Enriched. You take your choice, okay? But if you are looking at some grade 11 courses, let me know, I can give you more information.
Right, so our neighbor, they're just going around. Yeah.
So folks, your homework, should I just give you 10 minutes to work on your homework? Yes, pages 5 to 10. Let me erase this and write it up here. If you didn't do pages 1 through 4, you also need to do that. If you decide that this is going to be a good time to fall behind on your homework, that would be a poor decision. You will not have quizzes this unit. You are doing homework to check your understanding, and there are no quizzes. And you're making your study sheets. If you choose not to do the homework and not to spend time making your study sheet and studying, then you will do poorly on the test. I do not want you to do poorly on the test. That would make me sad. It would make you sad. There would be many tears everywhere. Don't make me cry, okay? Don't make yourself cry. You're also at some point going to want to start collecting your vocab words into a list so you have them all in one place. I'm starring them and highlighting them as we go along, but you're also going to want to make sure they're all in one place. So you might even, if you want to, you could even use like the inside cover of your book. You could do it there. You could use a piece of paper, whatever you like, but start collecting your vocab words. You can put anything on there. You can draw pictures, you can lace diagrams, you can whatever you want. Definitions, whatever you want. Yeah. No. Science 11. Yeah. No, it's great. I'm not good. Something. Shut up. Something else, right? People think they're not getting science, and they're like, yeah, we are working on our homework, which is and you should probably So we're gonna look at this one. But now we're learning the yes, she can use your study sheet on the test. Okay? Yes, she can use it on the test.
Protein, yeah, and then protein, and then cells. Yep, and then protein.
All three, like, console called and not called. So it is a bit like.
Wait. That's crazy.
Bye.